So shall I start right
sure.
away?
Yeah, it's your floor now.
Okay. So it's a pleasure to be here. Thank you very much for joining. I would like to talk about butterflies and specifically, butterfly dispersal today. As you can see here, unfortunately, I don't have a presenter, but it should work anyway. I don't know why it does not. It does. All right. So let me start with introducing to you my general research interests. I'm generally interested in responses to environmental change, and those responses can be divided into three different types. There might be plasticity, plastic responses to environmental change, or genetic adaptation to environmental change, or finally movement patterns. And I'm interested in all those three strategies. I'm interested in the prospects and constraints of those strategies, and the aim would be to better understand the limits to population persistence and to inform modeling and especially conservation measures. But today I will focus on movement, on dispersal, but to also at least give you a flavor on the other topics, plasticity and genetic adaptation. I will briefly give you one example on heat resistance and the expression of heat shock protein. In this butterfly, you will hear about a little bit more today. It's one of my favorite butterflies, the sooty copper. And what we've done here is we compared replicated populations from high and low altitudes in the Alps. And what we investigated here is heat knockdown time. So this is the time you can measure until the physical knockdown of an individual. So the longer the better. You expose them to a severe heat stress and you simply stop the time until they will be physically knocked down. So the longer the better. The longer heat knockdown time, the better they can stand the heat. So what you can see here in this graph, so replicate high and low altitude populations which have been kept at either 20 or 27 degrees, you see the difference. If they have been kept at the higher temperature, they have a longer knockdown time. So this is a plastic response. If they are exposed to higher temperatures, they get more heat resistance. You can also see, if you compare these two, for instance, the genetic differences. So this is a genetic difference between high and low altitude populations. And what you can see is that the low altitude populations show a higher heat stress resistance. However, what you can see in the first place here is the difference in plasticity. So the high altitude populations, they show only, rather limited plasticity, whilst the plastic response in the low altitude populations is much more pronounced. And this probably means that those guys living in high altitudes of the Alps, they have lost probably their adaptive, their plastic capacities because they never needed it. And that might be an ecological trap with climate change. Having found such a pattern, we can now try to dig into the underlying mechanisms. And what we've done here is to measure the expression of heat shock proteins in exactly these treatments. And what you should see is that the pattern is exactly the same. So we find a much more pronounced upregulation of heat shock proteins in the low altitude populations kept at a higher temperature. So it's exactly the same pattern, which does not necessarily mean that there is a causal link. It might be a correlation, but at least it's an indication of the underlying genetic and physiological mechanism. All right, so this was a flavor on plastic and genetic responses. And now I will switch. So that's the key message, low plastic capacities in the high altitude populations. And then I will switch to dispersal, and I click a bit further. So the first question is, of course, what is dispersal? And just a definition, it's any movements potentially leading to gene flow. So as you can tell from this definition is that movement and dispersal, they are intimately connected with each other. but it's not connected with migration, at least not directly, because migration is a directed process, whilst movement or dispersal is not directed towards a defined like south-west in our migratory birds, and it's typically back and forth. So movement and dispersal are connected, whilst migration is something different, and it's a complicated and it's a complex matter. And why is that? It's because it serves a variety of different functions and because it's affected by different external as well as internal factors. So let me first talk about the importance. So what different functions? One function might be foraging, which we just did. So if you are in your flat at home and you don't have anything to eat and you are hungry, you will go foraging. You will disperse from your flat to a supermarket, to a restaurant, wherever. That's one function of dispersal. I'm reluctant now to again talk about humans and main location, but anyway, it might be predator escape or avoidance. It might be to explore new or better habitat or reach new or better habitat, or to maintain metapopulation connectivity, to track shifting ecological niches, which is very important for less mobile species, and it's very important in times of climate change. So it's a key biological process we are talking about here. And often the question is, should I stay or should I go? And now it is affected by a wealth of different factors. On the one hand, we have the internal factors like condition, body shape, flightability, the morphology, but also the motivation, the personality. And then we have the external factors, which include the environmental context that can be abiotic or biotic. And the external abiotic factors might be physical barriers, like the sea, the large lake, or a mountain ridge you are not able to cross. That's physical barriers. It might be influenced by climate, by weather conditions. And then, of course, you also have the external biotic factors like competition, diseases, resources you need. And all these different factors may facilitate or they may constrain dispersal. So to sum up, dispersal is a very important part of a species' resilience to environmental change. And this is especially true for so-called sedentary species, because they have problems with reaching new habitat, because typically they stay at home. So let me outline my talk. The good news is you survived the introduction already. Done. The next will be Margulies recapture studies, so the more traditional approach to dispersal. I will talk about condition and flight performance, to small scale, I mean really small scale movements, then a bit larger dispersal propensity, then factors facilitating range shifts, a little bit about landscape genetics, then resources and range shifts, and then I will wrap up my talk and give you a brief outline. So one done, but there is a lot more to go, so I hope you will enjoy it. So let me start with Margulies recapture studies, and I did it with this beautiful butterfly, the violet copper. It's a post-glacial relic species in Central Europe. I don't know whether you have it anymore in the Czech Republic. As far as I know, it's gone, right? It's a habitat specialist. It's monophagous, just one plant it can use, and it's considered to be extremely sedentary. So we are now somewhere here, and the study has been conducted in Western Germany. This is my home area. And this is again my home area. You see here the Westerwald mountain range. And also in former times, it has been always restricted to the largest mountains and ridges of this area. And this is not good news. Because the mountains are not very high and they are sitting on the top of the mountain. So with climate change, they are really in big trouble, right? It's not like in the Alps where at least some species will be able to climb up. This is not possible in these low-range mountainous areas. A male and a female, still extremely beautiful, enjoy it. And now it's going from the top of the mountains down to Earth. So this is what we found roughly 25 years ago. So this is each dot is a population or a former population in my home area. There were around 120, and see what happened with over time. So from what was 110, we are now down to 30 populations. So an extremely strong decline in population numbers within 25 years. And of course, one question is why? And we then did a mark recapture study. And you can see one of my study sites here. They occur here in this gray shaded area. So here and here and here, there were 3 patches, and each arrow indicates inter-patch movement. So there is quite a little bit, you see the scale bar here, there is quite a little bit of movement among patches, but not a lot. And indeed, what we found is that the average dispersal differences, the distance in males was only 37 and for females only 60 meters. And the maximum was 560 meters. So this is really considered, I mean, these average values, even for mark recapture studies, it's very low as compared to others in individuals. So this sort of proves it is likely a rather sedentary species. And in accordance with that, we looked at patch occupancy, and the factor we found which had the largest predictive power for patch occupancy was the mean distance to the next 3 occupied patches. So habitat networks, obviously because they can't travel for long distances, are very important for this species, and to a large extent predict the occurrence in a given patch. All right. So that were some early studies on one of the copper butterflies I'm working with, indicating indeed they do have a largely closed population structure and that they are largely sedentary. We then, using the sooty copper, did experiments on condition and flight performance, and it's sort of fun to do this. We have the so-called shaker test. So this is an ordinary lab shaker. We put a plastic bottle on top of it, let it shake, put a butterfly in, that's all. That's really sophisticated. I would like you to adore this sophisticated approach. So you release the butterfly here, and it's shaking like hell, and it will try to fly as long as it can, because otherwise it will lay down here and the wings will be ruined, etc. So as long as it can, it will fly there. So this is a test for flight ability. And we also did the tunnel test. So this is a plastic pipe, and there is a light at the end of it. And here in front, you release the butterfly and you just look whether it's willing to approach the light source in a completely dark room or not. And this is considered a test of flight willingness. Those were the 2 tests we performed. But first, let me introduce the sooty copper to you. You see, it's also a nice species. If you disagree with me, please go now. I really love these things. It's a widespread temperate zone grassland butterflies, which is nowadays often, not everywhere, but often declining due to agricultural intensification. It's a sedentary species also, relatively sedentary, with close population structures. Nevertheless, it has shown a range expansion towards the northeast in recent years due to presumably climate change. Here we go, shaker test. So we tested for flight ability. Here you see flight endurance, and we did it at 2 different ambient temperatures. Higher temperature, better for the butterflies, they can fly for longer, that's trivial. But what is interesting now is food resources or no food resources. So part of it will start for 2 days before this test, and the other one had full access to food. And you see that starving the butterflies reduces flight performance. Trivial result, expected result. Then we did the tunnel test. And here you see First, for males and females, you see generally males have a better flight performance, whatever we are testing here with the tunnel test, but sort of flight performance, they are more exploratory. And again, control and staffed ones. So you see again a reduction in the staffed butterflies, and this is sort of strange, because if you don't have to have anything to eat, you should be more willing to fly, I would say, to go foraging. So this is a little bit of a strange result, I would say. And what does it tell us? So first, what we could show, that larval as well as food stress reduced both flight ability, shaker, but also the willingness, the exploratory behavior. Males generally have a higher dispersal ability than females. I told you it's a little bit strange, so we are not sure anymore what this tunnel test is actually testing for. But then the question is, I mean, you shake butterflies in a plastic bottle. Wow, that's cool. I mean, it's really sort of cool, but what, if anything, does it tell you about dispersal in the wild? I can't tell you. But at least we wondered about that, and we were wondering about whether good shape makes good dispersers, which I think is a straightforward assumption. So if you're in good shape, if you're fully fat, have a lot of fat reserves, you should be a good disperser. So now we go from the bottle to the next larger scale, small scale movements in the same species, and small scale means within the climate cell. What we did here, 5 by 4 meters, 20 square meters climate cell, we had here release cages, and then we had different squares in here with some flowers for feeding, etc. And simply every 30 minutes, we located the position of each butterfly, and we simply counted the number of different positions. So some were sitting all the time here, and others were moving around. This is what we did here. And what I would like to point out, if we just compare females and males, what do we see? We see females are heavier, should not be good for flight, especially they have a heavier abdomen, whether the thorax is actually heavier in males. So males have a higher relative investment into flight muscles than females. And also the wing loading is higher in females. But now guess the results. At least in our climate cell, females are more dispersive than males, although they have, or should have, a reduced flight ability. This is also what the shaker test showed us, right? Reduced flight ability, still they move more, and if you paid attention to the first example, violet copper, you actually could see the same also in mark recapture. Females do move further. So, all right, still we were wondering, what does it tell us? Okay, we went from a bottle to a climate cell, should be an improvement, but maybe we can do even better. And then we went to southern France, which is a fantastic location to study dispersal. We went to the so-called Metatron. So all these are cages, and all these cages are connected by dispersal corridors. So this is what this, fantastic place looks like. All these are cages, and they are connected through these dispersal corridors. We used the most simple design you can imagine. We always connected 2 cages. So one release and one target cage connected by a single corridor. So we just wanted to know the dispersal propensity, the willingness to leave the cage where the butterflies are in. In the target cages, they all had the same conditions. All were sunny with loads of resources available, but the release cages, they differed. We had the good one, sunny, resources. So here actually, there is no reason to disperse. Then we had two, sunny and shady, and in both no resources, they should leave. And here we had resources, but no sun, it was shady. So now let's have a look at the results. We've seen a much higher dispersal rate in the shady cages, which is actually very interesting. This is really interesting. On the one hand, it's sort of expected, no good conditions, they should leave. On the other hand, shade does, of course, reduce butterfly activity. And this tells you, this is not just random dispersal, that it was an active decision to leave. No resources, much higher dispersal propensity, and again, the females are more dispersive than the males. This again, this is straightforward, right? This is what you would expect. And again, we can show also with these butterflies from the Metatron, which we afterwards subjected to our bottle shaker test. Those with no resources in the release cage, they had clearly a poor flight performance, but nevertheless, they disperse more. Woo. So, what we could show here is that the propensity to disperse, the decision to disperse, does not equate with the capacity to disperse. It was found, the propensity was found to be decoupled from flight performance and capacities, and other traits indicative of condition. So good shape does not necessarily make good dispersers. This we could show based on shade, like a food, females. So what we then thought is maybe it's motivation, maybe it's behavior that is dominating. And we thought, well, let's have a look at dispersal in leading edge populations. And we do need, if we really want to understand what's going on in the field, we do need genetic methods, because then we can also assess the rate of dispersal over longer periods of times and potentially also on wider scales. So, factors facilitating range shifts. I told you this guy is currently spreading northwards. And what we did here is we collected populations from core populations in northeastern Germany, and also replicated populations in northern Estonia. And these populations have been established like 2 years before we went there for sampling. So really newly established edge populations. And we were wondering whether the edge and the core populations would differ. We found no differences whatsoever in flight performance, along with what we found before, that dispersal is not necessarily linked to flight performance. But what we found is here you see the food reaching rate. So this is in an environment the butterflies don't know. How long would they need to find a food, which is a measure of exploration, exploratory behavior? You don't see much difference. So this is males, females, Germany, Estonia, and the whole thing down at 18 and 26 degrees Celsius. You don't see much of a difference at the lower temperature, a little bit of a tendency, but at the higher temperature, you do see that exploratory behavior is more pronounced in the newly established Estonian populations, which again indicates that it might be motivation, that it might be behavior, which is the key rather than morphology or fat reserves or anything else. All right, let's go now to the larger scale. Let's go to landscape genetics. And this is, I mean, I do love copper butterflies. I hope you understand. And I do love the Alps. I do love mountains. So we went to Austria, to the Ötz Valley, fantastic. And here you see our sampling scheme. So we have here the main river. Each red symbol is a sampling location. And we have 2 different tributaries. And all these dots, we collected butterflies, mainly along, what is it called? The river meadows, so along the river. But some, especially these two and these, also above the river, so just like 200 meters higher, which will be important. So what have we done? DD-right sequencing approach. We used roughly 13,000 SNPs if you compare to earlier times with microsatellites where you have maybe 10 or 20. So this gives you a lot of power. And one example I would like to show you is what we did is an estimated effective migration surface. So here you can hopefully still see our sampling scheme. And here are the colors explained. So basically what you see in this area, you get the expected pattern of isolation by distance, but here you do have a dispersal corridor. So this means that all these populations are very well connected with each other. And this is roughly 15 kilometers. And before, at least in the violet copper, we were talking about a couple of 100 meters. Now we are talking about 15 to 20 kilometers, where there is a very high connectivity, genetic connectivity between those populations. We then did a distance-based redundancy analysis with which you can test whether there are any dispersal barriers in this landscape. And this is the result. Here you see the different genetic clusters. So these in the black circles are genetically different from the others, whether those are all the same. So what does it mean? Let's start with one. Here you see those populations are isolated from all the other ones. Here there is a gorge with no habitat available for the butterflies. So this is likely a dispersal barrier. We see 6 and 7, again, gorge here, gorge there. So they are, well, not isolated genetically, but they are genetically different. Also, you can see the two populations which we collected not next to the river, but on the plateaus above the river. And also you can see these two are genetically different from the others, whether all these here along this area, they are genetically, you can't distinguish them genetically. We also tested for the impact of the road. So along the river, there is running a road. And then we just put all the ones left-hand side and compared them to right-hand side of the road. And we saw a significant genetic divergence just caused by the road. And now it's getting even more intriguing. If we do the same with the river, no difference. You explain me now. Imagine you're a butterfly this size. This suggests you have no problems with crossing a river, which is cold, which is wet. But a road, which is hot, which is excellent for flight, you don't cross. So what does it mean? Well, first, sedentary sooty copper is surprisingly mobile gene flow, at least for like 15 kilometers. Nevertheless, we could show that ravines, gorges, and steep wooded slopes comprise sort of at least relative barriers to dispersal. Roads, but not rivers, comprise barriers to dispersal. So it might be that the isolating effects of anthropogenic structures are more important than those of natural barriers. That's at least something we are thinking about at the moment. Yeah, and the SNP approach with these very high numbers of markers, yeah, it's just a fantastic tool to reveal even fine-scale population structures. All right, then resources and range shifts. And now I would like to draw your attention to host plants. Butterflies are herbivorous insects, of course, so the availability of suitable host plants is very important. And it's even more important for range shift. So what might happen? It might constrain range expansions. So the first question is in the new area, the primary host available in sufficient quality and quantity? If yes, no problem. If not, is it possible to integrate a secondary host? And if this is also lacking, then the question is a host shift possible? And if not, you can't expand your range because there is no suitable host plant available. So host shifting ability may largely determine range shifts in herbivorous insects. I now have to leave my beloved copper butterflies. But this is also a nice one. Here is money. It's a very special butterfly for reasons I will tell you in a minute. It has several generations a year. It's oligophagous. And until like 10 years ago, it was also considered to be very sedentary. Ooh. So what did this beast do? You here see the largely Mediterranean distribution of this butterfly. And I would like to draw your attention to this subspecies, Alpigena, because this did a really strange thing. This beast was the first time ever recorded in Germany in 2008. 10 years later, it reached the North Sea. So within 10 years, a range expansion of 1,000 kilometers to the north. It's quite a lot, I would say, for a sedentary butterfly. We did the same approach as with my copper butterflies. We collected populations in southern France, so in the core of the distribution area, and we collected populations in the new range, so within a couple of years, newly established populations. And we first looked at the female oviposition preference. We tested for each female 5 different plant species, and we did this for the French and also for Germany. So for French is core population, Germany is edge population. We tested all the butterflies, and the gray bars are the French females. This is the native host plant of the core French populations. And what you can see is that almost all the eggs were laid on the native host plant. So there's very little flexibility. So basically, they didn't pay any attention to alternative hosts. And now look at the German populations. What they did, they actually showed the highest preference for a plant which is not even occurring in France. So within less than 10 years, they do show a completely different oviposition preference in being much more flexible, using different plants to roughly the same share, 3 different plants, 2 of which also occur in France, and 1 of which does not even occur there. So that was fantastic results and interesting results, I thought, and this is indicative of rapid evolution in behavioral traits. We wanted to gather more information on that, and we also investigated then development time and cold tolerance. Let me start with larval development time. Here you see at 2 different temperatures. You see the French and the German populations at the higher temperature. We detected no significant difference, but at the lower temperature, which is of course more challenging, here you see that at the more constraining lower temperatures, the German populations, the German individuals developed faster than the French ones. So again, indication of rapid adaptation in edge populations in evolving shorter development times at challenging temperatures. And we also investigated cold tolerance, which in this case is the chill coma recovery time. That is the time an individual needs to recover from chill coma. So the quicker, the better. Again, those from the high temperature, no differences, but at the temperature at which cold stress is more likely, we again see a significant difference between French and German individuals, with the German ones recovering quicker from cold stress, which means they are better adapted to cold stress. All right, so again, rapid evolution in range expanding populations, which was great for us to see. I'm coming to an end soon. An outlook. I'm now so, actually I told you I was talk about dispersal, and I think I did, but I also talk a lot of genetic adaptation and evolution. And this is something what is intriguing us now in the context of agricultural landscapes at the moment. And we here use the small e. So this is the latest data we have, just to give you an idea what we thought. So this is just a conceptual figure of a modern landscape, which means you have large crop field sizes and you have only small fragments of semi-natural vegetation like field margins or meadows remaining. So the resources for butterflies are very scattered in the landscape, and this should indicate a traditional landscape where the field sizes are much smaller. You have a higher proportion of semi-natural habitats which are much better connected. And I can show you two aerial photographs. It's not good to see, but basically this is an agricultural landscape composed of like more than 90% crop fields. And this is sort of a traditional landscape. And our prediction was that in the modern landscape, the butterflies should be of poorer condition because they need to fly larger, and in combination there are less nectar sources available. But on the other hand, we expected selection on flight ability because resources are more scattered and they need to reach them, for instance, oviposition plants. Hot from the press, our very first examples, we so far analyzed the data of one landscape pair, so one traditional and one modern landscape. In each of the two landscapes, we had 10 sampling locations with roughly 20 individuals each. And we will investigate more of such landscape pairs. And this is what we found. For the first one, but it's very motivating, very motivating. This is thorax-abdomen ratio. So again, the investment into flight muscles versus reproduction, the abdomen. And you see it's as predicted higher in the modern as compared to the traditional landscape. So in the modern landscape, they do invest more into flight morphology, into muscles. And also what we could see is that in the modern landscape, they have larger wings. I should note this is not showing residuals, but of course we corrected for body size. They are exactly the same size in terms of total mass. It's only the wings that differ. It's not simply because they are larger. In body mass, they are exactly the same, no statistical difference, but still they have larger wings. So this is very exciting and motivating, and it does indicate that really modern agricultural landscape impose severe selection pressures on individuals. And I don't know whether you follow the whole debates about genetic adaptation in the current era of anthropogenic climate change. There's actually very, very little evidence that this is going on because it's believed that it takes so long that it's just not possible that species can genetically track climate change. I don't know what to think about, but at least if we look at differences in edge and core populations, or here in landscapes which are next to each other, like 10, 20 kilometers apart, but differing in structure, we do see evolutionary change. And at least in the other two examples, it was like within 10 years. All right, so take-home messages. We've seen that food stress reduces flight ability, but also the willingness to flight, which does not really make sense. So I still believe the tunnel test is actually crap. Don't do it. Flight capacity does not equal movement and dispersal propensity. And this is actually for all, I think, entomologists, bad news, because we also always use, as I did actually in my last example, right? Oh, they have longer wings, so they are better dispersers. We can actually show that there is no easy shortcut between the two. So we have to be very careful what we conclude from such data, because at least in many cases, I would say there is just no relation between the two. It's rather the detrimental conditions that increase dispersal propensity, which indicates that it's rather motivation than anything else. I've shown that leading edge populations show a faster development. We could show this in both species, the white, but also the copper. They are more exploratory, they are more cold-tolerant and more flexible in terms of host plant use. And we could show that this is very, very rapid evolution within at least partly less than 10 years, which makes you kind of wonder, how might this actually happen? So it's probably, well, maybe we can discuss, maybe you think about it and we can discuss, if you like, what might actually be going on there. We found that sedentary fidules, as well as other butterflies, can be surprisingly mobile, but still they are affected by barriers, as slopes and gorges. And natural versus anthropogenic barriers might differ. And also in relation to that, we think in the meanwhile that it's actually behavior rather than morphology, which is the key if you want to investigate dispersal. This is just for discussion if you want. So I hate green bridges. Don't know whether you think it's a good idea. So this is just a wolf which went back and forth between Germany and the Netherlands and crossed several motorways without any green bridges. So I think it's a waste of money. You can use millions of euros to build better green bridges. I would rather save red meadows with the monies, but this is something, if you want, we can discuss about. So finally, I would like to thank, of course, the members of my group, especially Elisabeth, Ronja, and Annika, three PhD students, which all did a great job. The German Research Council for funding, and of course, all of you for paying attention to me. Thank you very much. Open for questions, if you like.
Thank you very much, Klaus, for this exciting and fantastic presentation. So I think the stage is open for questions. I think there will be a lot of discussions around this topic. But yeah,
let's hope.
Nice presentation. I have a question about your two experiments with the Metatron and the one in the smaller scale. You said that some, like you were offering resources for them to, like some patches were rich in resources, some are poor. These resources are resources for adults, like nectar.
Yes.
How about host plants? Can this difference in movement between males and females be because females are looking for a host plant that is not there?
Yeah, we didn't look into that. So when there was a flower and I was talking about resources, it was always both. So in those cages, we had flowers for nectar feeding as well as host plants. So we did not separate the two. It was either no resources or all they need, both oviposition plants and nectar plants. So I can't tell you anything about the difference because we didn't test it.
Yeah, but the difference with the females, so it's accounted for, like it's not.
Yes, yes, and with the Metatron, you probably saw this particular shape of the dispersal corridors, so they don't know what to expect. They don't know what is on the other side. So it's really only suitable for measuring the willingness to leave the patch they are in, no matter what is on the other side. So it's really the dispersal propensity, the willingness to leave their patch, which is measured. Yeah. Basically, we didn't want them to go back. That's why all resources were there in the cages.
So I was really interested about the whole idea of you being able to distinguish the different populations in which how they are genetically different using SNPs. My idea is that did you also look into the patch size in relation to the gene flow? Like, because if the populations within each patch are dispersing amongst one another, I would have an idea that this patch size in itself also has some effect on gene flow.
If you're interested in gene flow, then you need the SNP. You mean the SNP study, right?
Yes.
We didn't investigate that. What we accounted for is isolation by these barriers. So isolation by resistance, these type of analysis, and of course isolation by distance. So we took into account the distance between the different sampling locations, but we did not look into patch size.
Yeah, no, I'm just coming from like more of like islands.
I'm just thinking. I think in this area it wouldn't be possible. So basically, at least in the one tributary where we did have lots of gene flow, it's basically more or less continuous habitat. So it's not like you do have different suitable patches, but you have the meadows just along the river on both sides. So I wouldn't know in this particular landscape. I think this would be much easier in a landscape like around here, but there I couldn't think of a way to even determine what patch size is.
Yeah, I mean, it's really hard to measure. So.
Yeah, so sorry, can't tell you anything about that.
Maybe related to that one, I was just puzzled that it was like two rivers, right? Like this branch. And one branch was like really well separated populations and the other one was continuous. So like what was the difference? So one river got no breaks in the habitat patches and the other one got breaks. Yeah, this one. They were like mobile.
Yeah. So here we had lots of gene flow, and these, oh, I forgot to mention it actually. So this is indicating potential barriers, and you see there is just one here, which is a very short gorge. So apparently they can cross it. While here you can also tell from the gap in sampling. So we really tried to sample everywhere. Here in this area, we couldn't find any suitable habitats. That's why here's no red dot. The same is here. So I think it's just because there are no natural barriers here in this area, whilst here we have a lot of different gorges, which are much longer than the one which is found here. I think that explains the difference.
And then you mentioned the road effect.
Yeah.
The road, where the road was based on the map.
It is always, it's actually indicated here, this thin black line. So it's all the time running along the rivers, and the same is here.
Interesting.
So it's just next to a river, and just you see, you do left and right of the road, you see a genetic difference. You do left and right of the river, you don't.
So there cannot be any related effect to these barriers, like, I don't know, they built the roads and distribute the habitats more on the both sides of the roads, while on the river they are more close or something.
No.
Okay. Interesting.
It's very strange. They are stupid. But I again think it's behavior. They are more reluctant to cross something they don't know. It can't have anything to do with flight ability or conditions, because the road is, of course, much warmer, much better for flight than a cold river.
Does it have heavy traffic?
Well, it depends. I mean, it's the Earth Valley. There is, in terms of domestic cars, not a lot, but tourists. So in summertime and on days with nice weather, there is of course a lot of tourists traveling, but this is then during daytime, which is the time when the butterflies are flying around. So yeah, maybe it's the traffic rather than the road itself. This is something we want to look into with these, for instance, edge crossing behavior. So we want to see whether if they approach a road, it can be somewhere here, it can be any road, whether they are more likely to turn around if they hit a road as compared to a river. This is something which is on the schedule for the future.
You know, like I was thinking if it might be something related to the color, because river is, well, is reflecting the sun and so on, but the road is black. So if that can influence that they don't cross, because if you change the color of the road, that will change. I don't know actually how butterflies behavior, like if it's more by odors or by colors. No.
I don't know, but well possible. So yeah, I think it's something in perception and behavior. Can't be something physiological in terms of flight.
Interesting.
Did you look for the story about France and Germany, the current edge? Did you look at the plasticity of the edge group? Like, does the individuals that are more dispersal, that disperse, expand the range? Are they more plastic than the core population? Or?
It was. Should be the next, right?
Yes, this one. Yeah,
because we also include temperature, you can actually tell plasticity from here. And because here you don't see a difference, and here actually it's that the German populations show less plasticity in development, but more plasticity in cold tolerance. So we went to all traits. This is just two examples. I think we measured around 10 traits, and there was no consistent pattern. So you can't tell that the one are more or less plastic than the other. There was just no consistent pattern.
If you look into genetics of this study, if like the core population is genetically more or less diverse than the edge population?
Not yet. Not yet, but last year it was a bit difficult because of Corona, but we sampled along the expansion route. So they are all in the freezer currently. They are also sequenced, so we are at the step of SNP calling at the moment. So it's in progress. In a year, I come back and can tell you the results. Yeah, because I mean, it's so rapid, this adaptation. It must be either, does anybody want to ask the question? Otherwise, I continue. It must be either, I mean, it's genetic, we can show based on common garden experiments. It is a genetic difference. So it must be either genotype sorting, because in situ selection can't be that fast. It must be either genotype sorting, that it's only specific genotypes that reach Germany, or what we also speculate about, that there might have been a hybridization event between two subspecies. And that the hybrids are the ones which are now the dispersive ones in this believed to be sedentary species. That's what we currently think, but with the genetic methods, we will be able to sort it out with any loss.
Samples of other subspecies.
Oh yeah. We sampled the whole of Europe. We have from the whole European range, but especially dense along this expansion route. 1 year more.
Any more questions?
No. Yeah. Following the last comment, why would you expect the hybrids to be more dispersed if they fit in these two populations?
I have no idea. No, it's speculation. It's speculation. But I mean, at least in general terms, there are some cases of this hybrid vigor that for whatever reason, the hybrids can be fitter than the two former groups. And one example related to dispersal is the, oh, it's not my animals, spider, the wasp spider, right? Which also like 2-3 decades ago has shown in Central Europe a fantastic range expansion. And it was actually way before we had severe climate change. People said, well, the wasp spider is invading and it's due to climate change, but it was actually early enough. And in this species, genetic studies could show that this is due to hybrid vigor. So the Western and Eastern population met somewhere in southern Germany in this area, and those were so fit, they spread it like hell. So there is actually in spiders one such example. There was another question somewhere there.
Yeah, it was more of a curiosity. I just wanted to know if you had any plans, because in the beginning you talked about this HSP, or the gene that was upregulated in the
Feature proteins.
Yeah, exactly.
Yeah.
If you have an intention or any plans to knock out the gene to try to understand a little bit more about. No.
No, it's hell. So first, I'm not a molecular biologist. I love landscape genetics because it gives us so fantastic tools, look into question we couldn't even ask before. So I think it's fantastic, but I'm not able to do it. And in butterflies, heat shock proteins is hell, because to establish it takes a lot of time. So basically what we used is Drosophila antibodies, which does work in butterflies, but they hit both the cognate and also the upregulated ones. So that's why our baseline is never at 0. It gives you a lot of uncertainty, and gene knockdown that would be beyond my expertise. So I gave up on heat shock proteins, also because so many people are working with it. And if you, then I would go for Drosophila, and I wouldn't do it in butterflies. I would see no advantage. Why to pursue this in butterflies?
So somewhere I think in the beginning you mentioned that the flight performance was decoupled from the dispersal propensity. So does that mean that the flight willingness was also decoupled from the dispersal propensity? Does that mean?
Yes,
Mean that?
at least in terms of this tunnel test. But as I told you, I would never use it again. So, because I'm just not sure what it's actually measuring. And I think it's so artificial, I just wouldn't do it. But in terms of the results of the tunnel test, it's true. But yeah, and also in the propensity, in the Metatron, it was also not related to condition, right? The staffed ones with poorer flightability showed nevertheless a higher propensity. There it was also decoupled, yeah.
Maybe last question. With these new methods like SNPs, rat sequencing, and so on, like it's very fantastic. Like you can see, like you can measure, like like gene flow in real time, like population extraction and so on. But do you think it's widening the gap between basic research and actual applied conservation efforts? And because maybe the understanding between these fields are like really separating each other, or do you think it's like closing the gaps between these two fields with these new genomic methods, for example?
You mean between
more
more
like real conservation actions?
Conservation and science in terms of molecular
like this
biology
basic
or
population genomics stuff, because until now it's like separated fields, but do you think with these genomic methods is widening the gap or closing the gap between these fields?
Yes, I think so, but why? Why do I? Well, yes, I think so. then I would need to come back to the green bridges. So what is the scientific evidence that we need green bridges? And for what species? Usually it's used by deer. Deer is not threatened. So why would you build a green bridge? So I'm very doubtful about that. And I think landscape genetics can give us new insights, but also in terms of the connectivity that is needed in the landscape. So I'm not a big fan on population connectivity. I'm rather Jeremy than Chris Thomas. I would always go for the managing habitat patches and the quality within patches to increase population sizes. That would be my preferred approach to conservation. And I would currently spend much less effort into the connectivity, but there might be some species which depend on it. But then conservation would be evidence-based, because then you would be free to decide, okay, this is really a species with really limited dispersal ability, unlike not the wolf and the deer. They are not limited. But there might be some such species, and then you can establish targeted conservation measures for specific species which actually benefit from it. That would be my idea. I think basically green bridges are serving the companies. They are built by the same company who are building the motorways. So they actually earn double, and I think it's horrible.
I have one question.
There is a question from the off. Yeah, please go ahead.
So it's kind of following on, really interesting talk, thank you. And yeah, it was kind of following on from Pavel's question before in terms of the connectivity. And so do you think, for example, with the outpopulation of the copper butterflies, clearly there's like these separate populations due to natural factors such as the gorges, but having these barriers for dispersal such as artificial or like man-made influences such as the roads or something, do you think that's going to be problematic for these populations? Or do you think these populations will be sustained in such a small, being like these small disconnected populations?
Yes, thank you very much. I think currently it's no problem. We are really talking here about very subtle genetic substructuring. So basically this is a matter of power, right? So with the 13,000 SNPs, we can show significant differentiation, but it's very subtle. So in other words, there is limited gene flow among one and the other side of the road, but there is. So gene flow does exist, but it's limited. It's more limited compared to other circumstances. But I don't see that currently, at least not in the context where we have looked at it, that this is currently like a big threat to population survival. That I think you can't conclude from this data. Is that all right? Does it answer your question?
Yes, thank you.
Thank you. Sorry.
To your first example, where you were in the slide, and we're talking about the reduction of occupied patches by the butterfly. Is it because the patches are like extinct and not getting recolonized, or is it because the patches are disappearing?
No, the patches are there.
The patches are still there.
The patches are still available. Yes.
Anymore.
Yes. But so. Yeah, I don't have the graph, but it's simply, so anything else being equal. So we're talking about the same patch quality, we correct for that, but then the likelihood of the more isolated patches, the likelihood of occupancy is much lower as compared to welcoming ones. But the habitat quality does not differ.
So nowadays you have the same number of patches as 25 years
yes.
ago?
Well, no, not 25 years ago. So this is due to habitat loss. This decline is due to habitat loss. So a lot of them, those patches do not exist anymore because they got overgrown by bushes or afforestation, or because of intensification of grassland use. So this is due to habitat loss.
So this is the number of actual patches, not of occupied patches. This bright graph.
This is the number of occupied patches.
But you haven't collected data about all available patches in each year.
Exactly, yeah. So this data here this is only from 1 year. And in this year, we only considered suitable patches of same quality. But this is only for one specific year. It's not over time.
All right, I guess, yeah, we should stop the discussion now. And it's very thankful for your presentation. And yeah, I think there will be chances to still talk with you in the later after. I think Irene is organizing something. We can look for speakers. And yeah, if you guys are interested to talk more about butterflies, yeah, population genetics, conservation, you are very welcome to join. But yeah, let's give a big clap to Klaus. Thank you. Nice to meet you. Finally, stop this. Do you know how to stop? I guess just.